Stanford University.
It was easy to come here today because I've left gloom and doom weather back in the Northeast. It generally will snow this time of year, so it may very well be flurries when I land back in Rochester tomorrow night. But it's a joy to be here. It's a beautiful campus. Congratulations to a really top-notch CVI. I get the letters, or the newsletters, and I do look at them. I don't read them word for word, but I do look at them. I recognize people from time to time who I know. But you're really running a first-rate operation here. Stanford's wonderful. For all the trainees, you're in really good hands. But you knew that already, right? So it's wonderful to be here and to talk about CRISPR. I'm still talking about CRISPR. I got involved with CRISPR when I was at Cold Spring Harbor in 2012, and I heard Jennifer Doudna just up the road talk about, I never heard of CRISPR, and she sort of dropped the bomb, which was the science paper that was published like a month or two later. And we were all flabbergasted and just like amazed at this newfound technology. And what I want to do today is just tell you a little bit, for those of you who haven't been really following it, I'm sure most of you have, but for those who maybe are distracted with your own work, I'll just tell you a little bit about the origins, the latest in how we're applying CRISPR to make mice. All of our work, all the work I'm going to show you today is done in vivo in the mouse. We don't do cell culture work, though I do recognize the importance of using the cell culture work, particularly with respect to iPS cells. But all of the work that I'm going to show you today, and there are a bunch of vignettes, so I don't really have like one long cogent story, I'll be bouncing around, is all done in the mouse. So I want to start first with these guys. So most numerous organisms on the planet, bacteriophage and bacteria, and we really owe a lot to our microbial brethren, right? Because you think about it, a lot of things, molecular biology was born from work done in these organisms. So you think of Crelox from bacteriophage, think of phage display from bacteriophage, you think of the restriction enzymes in bacteria and so forth. And there's this like interesting thought that this is like microbial warfare going on, right? So I don't like to think of it in those terms. I like to think of it as sort of a homeostatic balance. So you have defense mechanisms on both sides of the equation. And obviously the bacteriophage are trying to just continue to replicate, continue to infect. And so one of the recent obvious revelations has been this CRISPR that's in bacteria, completely unique to bacteria and archaea, not found in no eukaryotes, just totally unique to prokaryotes. And then the repurposing of that really elegant system, and you saw the review, it just came out from Emmanuelle Charpentier. It's an awesome review. She just sort of brings us up to date with all of the microbial work, that whole adaptive immune system. Who would have thought, right? A bacteria with an adaptive immune system. But now it's just taken right off, right? So like starting in 2012, 2013, you just see now it's just blown away the other genome editing platforms. And now it's in popular magazines. It's in Jennifer's book that maybe some of you have read. It's even in a movie. I don't know if any of you have seen this movie, Rampage. It's a horrible movie, by the way. Even though The Rock stars in it, and I like The Rock. Come on, I mean, but it is the first CRISPR movie, so that's why it's there. So this leads me to this thought, which is this global CRISPRization. So I mean, really, fundamentally, CRISPR has changed life as we know it. I mean, people, the average person doesn't know what Taq polymerase is, but the average person is starting to know. They ask me, what is this CRISPR thing? I'm at the gym. What's CRISPR? And I have to tell them what CRISPR is. Agriculture, way ahead of the game now. Wheat that doesn't rot, tomatoes mushrooms that last longer on the shelf. But aren't they supposed to rot for a reason? Like they're old. You're not supposed to eat them. I'm not so sure about that. But entomology, so you think of dengue virus, you think of malaria. These things are going to be addressed with this technology. Somatic genome editing, probably, I know Matt Porteous is doing that with respect to sickle cell. The highly controversial germline genome editing. What I'm going to talk about today, though, is experimental genome editing in the mouse. So that's the focus of this talk. What I thought I would do is just sort of give you what I call, it's kind of an update for those who maybe have been dabbling or haven't jumped in because it's like, wow, where do I start? I'm going to give you a formula for making mice very easily, very swiftly, very cheaply, very powerfully. And I want to first start out with what I call two-component CRISPR. So in its simplest sense, it's just an endonuclease that by itself doesn't have any endonucleolytic activity. And then a single guide RNA that the Doudna paper, Emmanuelle Charpentier in 2012, really introduced to us with itself is a two-component system, a CRISPR RNA and then the invariant tracer RNA. And then together, this ribonucleoprotein complex surveils the genome very quickly, and this is a beautiful paper from Bob Tijan and Jennifer Dowden several years ago. Within minutes, this RNP will find its target in the genome. And I'd asked Jennifer, does that mean heterochromatin? They actually showed in this paper that even heterochromatin. And in our experience, and I'd be curious to hear otherwise, we have yet to find a locus that's not accessible with this CRISPR system. So it's just amazing. So once it surveils and it finds its PAM sequence with the Watson-Crick base pairing, it then undergoes these sequential conformational changes that Jennifer really elegantly published on to expose the two nuclease domains so that you get this perfect clipping of each strand of the DNA, 3 base pairs upstream of the PAM. And then, of course, the cell takes over, right? So then you just sit back and ask the cell to repair this double-strand break. And in the 2-component flavor of CRISPR editing, we have this non-homologous end joining process that occurs, which is, and I'm simplifying things here. For those who study DNA repair, I apologize. I know it's very complicated. There's resection, there's conversion. But there's basically rejoining because the double-strand break is incompatible with the life of the cell. And during that process, things happen, right? So usually deletions, but you can get insertions as well. But recently there have been some papers coming out, this one in bioRxiv, I don't think it's out yet formally peer-reviewed, showing that there's some predictability even with the 2-component CRISPR with a non-homologous end joining repair. And Eric Olson, I don't know if he's been here, but he's now going to cure Duchenne muscular dystrophy, and he tells me he has, with his editing, he gets a bias for 1 base pair insertion, which I didn't know existed. So, but apparently, if you look at this paper in bioRxiv, and there's another one, there are programs that are coming out to predict the outcome of this 2-component CRISPR repair. Now, if you add a donor template, right, then you have this 3-component CRISPR. And here is where it's precision editing through not, I don't like HDR because it's really not HDR in the sense that we think of it in mouse transgenics or mouse ESL targeting. It's really single-strand template repair. And it's, again, complicated. I'm skipping over, but I just want to be clear. I know people refer to it as HDR, but I like to call it SSTR more accurately. And then this is the outcome. Much less efficient than the 2-component CRISPR, but much more accurate. So here's some data unpublished. And by the way, most of the data I'm showing today is all unpublished. Some of it's very embryonic, so I apologize if it's not a complete story. But that's just how I roll. I don't like showing things that are, I think there's maybe one place where there's some published data. So in the early days, 2013, right, we used plasmid, didn't work. We used RNAs, that worked. And this is just data to show RNA versus RNP. So now it's quite easy to get these reagents. You don't have to do the in vitro transcription and the purification, the bioanalyzer, and so forth. We just buy it off the shelf. In fact, we buy it from Synthego, as you'll see in a moment, right down the road here. And you can see that there's a decrease in live-born burst. That was because we were doing pro-nuclear injections, and there's something about the Black 6 genomic architecture that's toxic. I'm hand-waving. We don't have data, but definitely there's a compromise in litter size, but the HDR efficiency is much higher. So now CRISPR ease. I call it CRISPR ease because it really is simple now. And I just rotated off study section. Honestly, if you're writing a grant there, you really should make the mouse or the cell line if you're doing cells because it's so doggone simple. So we get our single guide RNA. We design it through CRISPR. There's a lot of programs. I love Max Hausler's CRISPR program. There's the URL. And just, by the way, I always leave my talks on the computer. So for those of you who want the talk, you can have it. It'll be here, so for things like the URLs and things that I might zip by fast, you'll be able to capture it. So CRISPR we use, and you'll see another reason why we use it in a moment, to get the guide RNA. And then we just order from Synthego. The Cas9 protein is like $100, and the guide RNA academic discount, I think, is like $180. So very, very inexpensive. The oligo, of course, my EDT if it's 3-component CRISPR. So literally, from the moment we get the reagents in hand to the handoff to our core, because we don't do the injections, we have a really capable core, in 4 weeks we can get genotypic data, which is fantastically phenomenal, right? So here's some considerations before and during and after CRISPRizing mice. So genotyping strategy is very, very important. And I'll show you some of the strategies we've developed to even detect single nucleotide polymorphisms in the mouse. Microinjection is the sort of the standard that most of the cores use, but lately electroporation has gained some ground. And in fact, there's a new method called, and I'm sorry, it's called GONAD, which is a horrible acronym, but that's what it stands for. And we're actually trying to develop this because this obviates all the pseudopregnant females, the stud males, the microinjection, you just do the electroporation in situ in the oviduct. So embryos are in there, you inject the components of CRISPR and then electroporate and you get edited embryos, which is fantastic for things like whole genome sequencing for off-targeting. It simplifies things of that nature. So we're trying to get this to work. I'm told it works. It's an electroporator for 15 grand, and call me in 6 months or so and ask me how gonad's coming, or maybe more correctly, how the oviductal transfer is going. All right, so the other thing to consider is off-targeting, and I've been sort of up and down with this sort of issue from the beginning. What I love what Max did with CRISPR is he's got this. So here's a screenshot from CRISPR, and you can see that where the hand is there, that we've got a window that comes up, and has this got a pointer? Yes, it does. So you can see here are your off-target sites, and then there's this little piece of information here, which for the mouse is very important to us because it tells us that this off-target site will likely be bred away with mice. Not true in cells. It's one of the issues I have with cells. They're indelible off-targets if they occur. But in the mouse, if it's not an LD with our on-target edit, we ignore it because it will go away. And then finally, mosaicism. It's a reality. With RMPs, that issue is reduced dramatically. But we genotype, as you'll see in a moment, we genotype around that. We don't really care about like big deletions and that. We just genotype for exactly what we want. And we've gotten just about every project. And we've done over like 30 mouse strains. So I'm not going to show you all 30 mouse strains, by the way. So I'm going to take you now through, and this is really the bulk of the talk. I'm going to take you through some vignettes, some just short stories of how we've used CRISPR in the mouse to answer some fundamental questions that really were not impossible, but were risky, let's say high risk with the old way of doing things with ES cells. Most of my lab is interested in dark matter, so that would be the 99% of the genome that we have. We're gaining insight about it. Thanks to ENCODE, and I don't know if there are any ENCODE investigators here. I know Mike Snyder's group's here and have really owe a lot to them for the work that they've done over the years. These were the 30-odd papers in 2012 that came out in these 3 journals that really informed us that most of the genome is actually functional, and you can define functional in any number of ways. But for us, and for purposes of this talk, we're most interested in those functional sequences involved in the regulation of gene expression. So we're talking about enhancers, we're talking about transcription factor binding sites, which number in the millions, and we're talking about long non-coding RNAs, which I don't have time to talk about today, but I can talk to you privately about it. So we've got a whole battery of human link RNAs and some interesting ways of studying those using back editing. Okay, so here's the transcription factor and its binding site that I want to introduce you to, and this is really what I'm going to be talking about for the rest of the hour. And this is a serum response factor. Some of you may have heard of it. It's what I call a first-generation transcription factor. It was published in the first sequenced and cloned in the early mid-1980s by Richard Treisman. And it binds this box, this element, this 10 base pair element called a CARG box, which I'll have more to say about in a minute. But if you've never heard of SRF, it's a widely expressed transcription factor. This is something I just stole off the internet, the GTEx portal, which many of you may be aware of, and you can see that it's expressed pretty much everywhere, but particularly you'll see like in muscle. So like this is vascular smooth muscle, and this is bladder, and then this is smooth muscle of the GI tract, and then uterus. So for some reason it's particularly abundant in these muscle types, but again, it's in every cell we've looked at, including cells like platelets, which don't have a nucleus. Incredibly, like a lot of transcription factors conserve. So across kingdoms, so you can see in Protista, really high homology in dictyostelium, in fungi, of course, animals, 100% conservation across. This is the functional domain of SRF called the MADS box. Amazingly conserved, and most importantly, essential for life. Can't live without it, so no Anne Hathaway if she has both of her SRF alleles inactivated. In fact, in the mouse, it's been shown, and presumably the same occurs in humans, that they have defective gastrulation. So you get like basically a ball of cells with a knockout SRF. Very important transcription factor. In fact, we know of, as of today, we've been studying this transcription factor since I was a graduate student 30 years ago. Ooh, I told you how old I am. 74 cofactors, 5 of which are shown here. All kinds of programs of gene expression. So imagine this transcription factor in every cell dancing around the genome and landing based on the cofactor it interacts with. And so these are some papers and unpublished data of ours where we've shown, for example, most recently in the kidney and the podocyte, loss of SRF, the podocyte, basically the cytoskeleton goes awry, and the animals die within 6 weeks of acute renal failure. In fact, with the exception of adipocytes, every knockout of SRF is lethal, even conditional knockouts. So it's a vital transcription factor. Now, the function of SRF has been studied for decades, but the interesting point here is the regulation of this factor is virtually uncharted. So there's two whole papers that addressed the regulation of SRF in vivo. So again, my lab's mostly confined to in vivo model systems. We do a little bit of in vitro, but most of the work's done in the mouse. And these are the two papers. So one paper described some elements in the 5' promoter region, and the other paper described a TBX5 binding site in the 3' UTR. Now, again, here's a half of a screenshot, you'll see the other half in a moment, from the UCSC Genome browser, and we thank the ENCODE people again for contributing all these data. We can use data from ENCODE to sort of guide us as to where things might be, and you can see in the first intron, second intron, there's some business going on, of course, in the 5' promoter with these different chromatin signatures. There's suggestive data for binding. Now, I know it's busy. I'm going to stay here for maybe 25, 30 seconds. So for a long time, we've been very interested in Vivek up there. I see him. He remembers this when he was in the lab. Very interested in annotating all potential binding sites, what we call the cargome, every carg element in the human and mouse genome. And so what you see in the top track, in this special track of the browser, is what we call the cargome. And the red carg boxes are conserved. That means they're conserved in all animal species. Okay? We've, on top of that, done ChIP-seq. And so the next two tracks are ChIP-seq and human coronary artery smooth muscle cells, and ChIP-seq and human umbilical vein endothelial cells. And so this informs us as to where SRF is binding. Now, I put this slide in because TQ, who I see, had queried me about this locus. And I said, oh yeah, as a matter of fact, we are looking at this. And so this is one of our grants, is to study this transcription factors regulation. So what regulates the regulator? So we have all these data, including data from ENCODE here, you see right here, that corroborates the data that we have from our own lab for these carg boxes that regulate SRF. Now remember, I told you SRF binds a carg box, so this is sort of like an auto-regulation of itself. So, but here's the question. Are these elements really important? Are they necessary in an in vivo context? So I'm not telling you the several in vitro papers, because again, we don't care so much about in vitro data, that these carg boxes are functional in a dish. So you can do a luciferase assay, show that SRF binds and activates, so on. But are they necessary in a mouse? So here's the first edit. This is a 3-component, so 3C is 3-component CRISPR. So right away you're thinking, okay, he's got a donor template. You see that in the bottom. And this is the schematic I use. I hope it's clear. So there's the locus. There are the two carg boxes in the upstream promoter region. And here's the wild type sequence. There's carg box far, and here's carg box near. That's the double strand break. There's a PAM sequence. And then here is the donor template, right, with some substitution. So we're not a huge fan of deletions, though you'll see some in a moment. We like subtle editing in the mouse. And so you see several base pair substitutions. The other thing you'll notice is one of our genotyping strategies. So this is beautiful. So how do you genotype these mice, right? So it's very straightforward. You just make 2 forward primers, 1 with a tail. And then you can resolve it right on a 2.5% gel. So you can see here's the wild type band, there's the mutant band, and you see all varieties of genotype in this particular case. So now, are these carg boxes previously shown in vitro, are they really important in vivo? And so the answer is, well, maybe. So there's a little suggestion of maybe a decrease, but these animals live fine. They're happy in the cage, the homozygous mutants. So we move on. And so we move on now to this second intron with more evidence of SRF binding to carg boxes in this second intron. And here, and there's activation. There's one of the few in vitro studies we do. So here's the data. And so TQ, this is for you. Probably we'll collaborate. We'll talk. So here's one of the few times we'll do a deletion. So we're using 2 guides, 2-component CRISPR, but we're using 2 guides, Cas9, to clip out that region of the second intron that has the carg boxes right there. And then you can see we have 3 founders. These are hot off the press. See, they're red. They're hot off the press. And of course, we've got to breed them to germline transmission and then homozygosity. But don't be fooled. I don't believe that these are biallelic targeting events, because, as many of you know, small products are preferentially amplified over large products. So I don't think these are biallelic, but these animals are alive and well, and we'll be studying them. Now, while we did that, we also made a subtle edit of this same carg box. And now here is an RFLP assay where we created a SAC2 site, and now we can detect the products of a SAC2 site when we PCR around it in these founders. And so we're really excited now to study these mice to ask the question, are these carg boxes in isolation and in the context of the upstream carg boxes, do they really impact SRF expression? So what about myocartin? So I want to introduce to you now one of the 74 cofactors of SRF, and this is myocartin. And together, the myocartin SRF carg triad is necessary and sufficient to make smooth muscles. Think of MyoD in skeletal muscle, and you get myocartin in smooth muscle. So the only paper in vivo published on regulation of myocartin is from my postdoc advisor, Eric Olson's lab. This is more than a dozen years ago. You see that they described, in this case, it's circled right here. This is a putative enhancer, about 25, 30 kb upstream. They clipped it out, hooked it up to LAC-Z, and they got beautiful cardiovascular activity of the beta-gal report. But again, question, is it necessary? So when we did the experiment, we clipped out that putative enhancer, much to Eric's disappointment when I told him. He was like, oh, really? Darn. Not necessary. I mean, the mice are fine. They express normal levels of myocartin and the link RNA that's between it and that enhancer. So then there was another potential enhancer upstream. Same idea here, 2-component Crispr, 2 guides, and Cas9. Got one founder with this 25 kb deletion, nada. No effect whatsoever. So now we have a situation where we have 2 enhancers, either one deleted in isolation, no effect whatsoever on myocartin expression. Meanwhile, we were doing 4C because we really want to understand myocartin regulation. And for those of you who do this, you'll recognize this schematic right away. These are contact points between myocartin, which lives on chromosome 11, and other chromosomes. Really intrigued about this, but we haven't tackled that one yet. But a lot of activity in here, as you might expect. So finally, my postdoc said, dude, why don't we just take the whole thing out? So that's what these guide RNAs show, and we're going to take the whole thing out. So it's about a 300 kb piece of DNA that we're going to just clip out. Those deletions are trivial with CRISPR. You might think, wow, that's a lot. But people have actually deleted a whole chromosome with 2 guide RNAs. So you do have to worry about inversions. So inversions, as the distance increases with deletion, you have to sort of genotype for inversions because those happen with high frequency. So, but these are very, very early days. I only show it because, again, TQ prompted me. All right, now, I want to go back to SRF, remind you that it binds this carg box. And I had a very talented bioinformatician who would be in good company here, who wrote some code to interrogate the entire mouse and human genomes for every permutation of a carg box. And so now I want to tell you about different versions of what we call a cargome. So in the first iteration, there were 60 new targets. In the second, there were greater than 5,000. The third, about 150,000. And this is human. There's a number of carg boxes on each chromosome, and then the number of carg SNPs. So we're very interested in carg variants. Okay? And the reason is this, because we published this paper several years ago, and we showed that if we modeled a carg variant, a real carg variant in people, some of you may have these, astonishingly, in some cases, abolished binding. This is a gel shift assay. Remember those a long time ago? So here's a gel shift assay, and you can just see, for example, this one, I don't even know what that gene is, but it's definitely binding SRF, super shift, and then the carg SNP really severely impedes the binding of SRF. So the question is, are carg SNPs consequential? Do they matter in vivo? And so now what I'm going to take you through now are a couple of examples of where we've made single base changes within the carg box. So here is our friendly UCSC genome browser again. Now I'm turning your attention to one of the target genes for SRF. Very well known. I cloned it in Eric Olson's lab. This is kelponin. It's the smooth muscle flavor of kelponin. You see it's definitely SRF dependent. You can see our cargome predicts some very conserved carg boxes with some encode data supporting it. And for reasons I don't have time to get into, we focused on one of these carg sequences, and we asked the question, if we make a single base change in this carg, remember before we were making multi-base changes, now we're talking single base change, is there any consequence? And the way we did this was, it was very systematic. And this is just some of the data, we wanted to know where the least tolerant substitution was across this carg sequence. And it turned out that this was one of them, okay? So this is a cold competitive gel shift. For those of you who've done it, you know exactly what I'm talking about. For those of you who don't, you'll just have to take my word for it that this is a cold competitor that doesn't really compete at all for radio-labeled binding of the normal element to SRF. Okay, so very poor competition here. So we decided to make this single base change, and very fortuitously created our friendly SAC2 binding, SAC2 restriction site. And here's the RFLP assay, very straightforward genotyping. And then we found that there was, with a chip PCR, as we might predict, very little SRF binding. But here was the kicker. So a virtual knockout, but in a tissue-specific way. So in vascular smooth muscle of the aorta, and presumably other vessels, there's vanishingly low SRF. And if we look at the protein in these immunohistochemistry, you'll gain that appreciation. So in visceral muscle, like an airway smooth muscle, for example, you see there's still kelponin there. But in vascular smooth muscle, we've effectively made kind of a conditional knockout or restricted knockout to smooth muscle. These animals have a mild phenotype, but we haven't stressed them yet because, again, it's early days with this. Now, when we did that experiment's about 2 years old, we were like surprised, first of all. But then we asked, well, is this like just a one-off? Like, okay, it happens in that context, but you're not going to see it again. So we chose this gene. This is the smooth muscle flavor of gamma-actin. There's 6 actin genes. And we chose this for a couple reasons, one of which is there's 4 card boxes in close proximity to one another. Statistically, that's not likely to ever happen. But these are functional, and it's been shown in other papers that they're all functional. And we targeted one. And we targeted the one that had a flavor very similar to the one I showed you in the kelponin locus. And we made the same substitution. There's the genotyping, a little more complicated. The duplex PCR we normally do didn't work so well. But again, here, another locus and a virtual conditional restricted knockout with a single base substitution. It was astonishing to us that that would occur. Now here's the latest version of the Cargome 4.0. So this is based on dbSNP, what is it, 151 now? I don't know, 600 plus million variants. And this is a collaboration, because I lost the bioinformatician, he left, he got a job somewhere else. And so this is with NCBI people, they know what they're doing. And so Di Huang has done this, and there's like now just south of 1 million CARG variants, which is like, it seems like 1 in 3.5 CARG boxes have a variant in somebody, which is amazing. So where we're taking this information, what I just showed you are proof of principle data. Those weren't real CARG SNPs. People don't have the C to G substitution I just showed you. We just wanted to provide proof of principle. Now as we move forward, we're prioritizing these CARG variants that are in people, and then we're going to sort of make these, sort of prioritize them based on whether they're in a haplotype block associated with some disease. And we're really agnostic as to what the disease is, even though we're a vascular group. As you saw earlier, we're in the kidney, we're in the GI tract, and so on. So that's where we're going with this. All right, so here are the take-home messages for this first part. Really, and TQ I know is really interested in this, others as well, need to understand what the function of these non-coding sequences are, because that's a lot of sequence space. There's a lot of variation there that will likely inform us as to disease causation. Some structural genes appear to be completely dependent on a single base and a single element. That, as a transcription factor jock, was unexpected. And it may be a trend. And I'm told, I had a colleague yesterday tell me that he has a link RNA, and that link RNA is CARG dependent, and the same thing. It's a tissue restricted knockout, only in vascular, with a very subtle edit to the CARG box. So it's not a one-off. Maybe it's a 3-off, but we'll do some more. Regulatory genes, perhaps not surprisingly, have redundancy built into them. And so it's not surprising to me that we whack on myocardial and SRF as we have, and we're not finding the kinds of things I shared with you with the structural genes. And finally, the CARG box is well suited to target because it's got a PAM at the beginning and the end. All right, so now I want to move into some coding data, and it'll go a little quicker now. So because most of our time is spent with non-coding sequence space. So there's a number of ways to go about this. You 2-component CRISPR, very popular. Just use a guide RNA, or as you'll see sometimes multiple guide RNAs, to create a frame shift and a premature termination codon, a knockout. But then also there's 3-component CRISPR where you can use precision editing and create with accuracy a premature termination codon. So let me introduce you to something that I know TQ's group is very familiar with. This is a new risk allele, a lyomodin 1 that is, for coronary artery disease. But the lyomodin gene family, I put this in last night because I didn't want to jump right into lyomodin and not tell you, what is lyomodin? Dude, tell us what lyomodin is. All right, so lyomodin is a gene family. These are cytoskeletal proteins. And they have different paralogs, as you can see. There's one in smooth, one in cardiac, one in skeletal. And this is actually from Vivek's JVC paper several years ago. Now, the lyomodin 2 and 3 genes were knocked out. And here are the phenotypes. So they're lethal. They're either late embryonic or early postnatal lethal in both cases. But the lyomodin 1 gene was not knocked out. And now I'm going to look up at Vivek and smile, because he spent about 1.5 years, right, with ES cells, never getting it, Sean. So remember, we were talking about sometimes you never get it. So and I said, dude, you got to graduate. So you got to move on. So, but then CRISPR came along, and he was rewarded. He did get on the paper. So we used 3 guide RNAs in this 2-component CRISPR experiment with Wei Yan in Nevada. This was one of the earliest CRISPR projects we did. So we really wanted to know what lyomodin did. So it created a deletion and a frame shift, and that's what you get with a frame shift, the premature termination codon. It's a true knockout. But then the phenotype, so we're vascular people, right, by training and according to the NHLBI. But then we had this like phenotype with this extended abdomen. I went, what the heck is that? And so it was very easy to phenotype, because you can see there's a lot of milk retained in the stomach. And these animals don't live beyond like 5 to 7 days after birth. So somebody, now this is important for graduate students, put everything in your thesis, okay? So I ordered Vivek, you spent a lot of time on that knockout strategy, put it in your thesis. That was a really good thing to do, because 2 years ago or so I got a call from the other side of the world saying, hey, did you make that knockout yet? Yes, we did with CRISPR, and we have this weird phenotype. So, well, we have a family with the same thing. And so it turns out lyomodin 1 is a new risk allele disease gene for this ultra rare. I call it ultra rare because there's only one family in the world that I'm aware of that has this mutation. So I had one daughter who passed in utero, and then the other daughter, that's not the daughter, by the way, and the other daughter lived like the mice, just like a week, and then died of this horrible disease called megacystis microcolon intestinal hypoperistalsis syndrome. That's a mouthful. That's even worse than CRISPR. And there's the phenotype, and it's just a horrific rare disease, a typical genetically isolated recessive disease, right? So consanguineous couples getting together, they're carrying risk alleles, and they unfortunately have these rare diseases. And there's a lot of them. And so in my lab, we're interested in rare events. We're not so much interested in GWAS. Apologies to the GWAS people. We're interested in rare alleles that have big effects. Now, while that was going on, because we were inspired by Vivek, of course, and Vivek had published this paper again in 2012 where he showed, and we don't do these experiments anymore, by the way. They're passé for us. Taking the promoter region of LMOD out, which has 2 CARG boxes, as you can see, in the promoter region, and putting it in front of the beta-gal and then asking, what is the readout? Well, we get very nice activity that's CARG dependent. Awesome. But is it necessary? So we did the same thing as before, right? So we're in one oligo, we're able to make these 2 transcription factor binding site mutations. And you'll just have to trust me that this knocks out SRF binding, okay? So this should be SRF dead, should not bind. And there's the genotyping, there's our duplex PCR, which is very powerful. Now, there's a little bit of a decrease in aorta, but at the protein level, almost nothing. So these CARG boxes are dispensable for LMOD1 expression. And obviously, we don't get the MMIHS phenotype. Now, before I move on to the next part, I want to just give you a cautionary tale, if I may. We thought we'd be clever a few years ago and say, okay, since we don't know what we're going to get if we use 2, 3, 4 guide RNAs and we make deletions, and let's make a targeted premature termination codon. So this is CANK1, and I know I don't have it labeled as CANK1. I'm not being coy here, but this is really bizarre. So it's a big protein. This is exon 5, and here is the guide RNA. There's the PAM, and here's the Cas9, and here's the repair template, okay, ... Now look at, we've put in the strongest stop codon. So the strongest stop codon, for those of you who don't know, is TAA. And there's a lot of literature about that. So we use the strongest stop codon, 2 copies, followed by an A for a frame shift. We thought for sure this is going to kill expression. Well, guess what? It doesn't. And so there's this, and if I don't know if Stanley's here, but he knows about CRISPR stop. I don't know if you've heard of that, but there's this new CRISPR tool called, or application called CRISPR stop, which just randomly uses base editing, which is a little different than what I'm describing here. It's David Liu's base editing. And I'd asked the question at Cold Spring Harbor, well, how do you know that these are really premature and you're getting a knockout? And they're not. So just be careful. If you contemplate something like we did, you may be in for a surprise. So for some reason, either there's an internal ribosomal entry site. I don't know. I'm not a translational guy. I'd love to hear any feedback from people later if you have any insight. But we didn't knock this out despite having a very strong stop. Okay, I love the fact that you have a clock there because I'm very sensitive about time. All right, so now I want to move to another vignette, and this is epitope tagging. Okay, now this is kind of hysterical. So there's our complex again. Myocartin comes in 4 different isoforms of predicted molecular weight there. And now here is something that I spent an evening once, and I just sort of said, let me just look at all the myocartin antibodies that are commercially available. And these aren't all of them, by the way, but these are most of them. And what you'll see, if you haven't seen it already, you'll see a duplicate, and that really ticks me off. So there's companies that buy from each other, so in case you want to see where that is. So this is the same as that. And they sell it, and antibodies are, what, 300, 400, some are even more. It's criminal. Okay, so the molecular weight in all these cases is 100, which is the predicted molecular weight. So Eric cloned myocartin, and then we got on it right away. So for 15 years, we've been in the dark with myocartin protein, like literally, like we have no idea. Now you look at this Western and you say, dude, that's an awesome Western. Look at that band. It's like one band. It's beautiful, right? No spurious bands. Well, but until we did this experiment, which is we cut the band out. Well, first of all, we couldn't knock it down, as you saw, if you're still with me. But then I said, okay, well, cut it out and let's do mass spec. Let's see if there's any evidence of no myocartin peptide to speak of. 0. No evidence of myocartin peptide. So this is worrisome, right? And meanwhile, there's scores of papers. I haven't counted them, but there's probably 100. By the way, I have to disclose that, and Vivek knows this to be true, that we have a supplemental figure with the incorrect molecular weight, okay? So guilty as charged as well. But my point is authentication, authentication, authentication. So we said, all right, well, let's enough of this. Let's just knock in flag, and let's see what the real molecular weight is. And here it is. So it's not 100 kilodaltons, even though predicted molecular weight would tell us otherwise. It's more like about 150-ish. And we know it's about 150 because MRTFA is about 150, and it's not here, but MRTFA, its cousin, migrates in the same place. So this is a Western blot, and you're probably yawning. But for me, I was really excited. This is the first real Western blot. Very little myocartin in the heart. That was a surprise. Eric called it myocartin because it's a cardiac transcription factor. Well, guess what? It's actually more of a smooth muscle transcription factor, as you can see here. Now, I'm immediately thinking, okay, this can't possibly be right. So Paul Herring, unbeknownst to me, he's at Indiana, was making had the same idea and used an HA MIC tag. And the molecular weight's the same. We've since made a 3x HA tag, and it's the same. So the molecular weight is 150 kilodaltons. I don't have the data because I don't have the time to show you, but just FYI, HA is the best for us. In our book, HA is the best. We like it a lot because, for one, it works on sections. Flag, and if I hear otherwise, I want to know exactly how you got it to work. Flag on sections does not work for us. And we've tried every which way possible, and flag antibodies, we bought several of them. They do not work on sections. HA does. So we love HA. Oh, and then the reviewer didn't ask us to do this. That would be a mean comment. But I said, you know what, let's just preempt the reviewer's comment, and let's do it anyway. Since we did the mass spec before, cut the band out. I mean, it's got to be there, and it is there, but not a lot. Not a lot. So, and not surprisingly, right? It's a transcriptional cofactor, very potent, so you don't expect a lot of coverage. We're really jazzed about this mouse because now we're poised well to do experiments we've wanted to do for a long time. Like we want to do ChIP-seq, we want to do RIP-seq for our linked RNA stuff, want to know long non-coding RNAs that are interacting with myocartin. And this is just some ChIP PCR from the paper that just came out showing that, yes, we can enrich for the usual suspects with a flag antibody. And we haven't done it yet, but I suspect HA will work as well. Okay, so you with me? So one more vignette, and this is the tough one, right? So these are the conditional knockouts. These are the larger things like putting in CRE and so on. So making conditional knockouts with CRISPR is hard. I'm just saying, it's hard. And here's why. So notwithstanding Rudy Yanish, his Cell paper in 2013. So the 2013 paper showed pretty convincingly, I think it was like 16 to 20% efficiency, that one could take 2 guide RNAs, 2 repair oligos, and Cas9. So I call it 3 component, but there's really 5 component, however you wish, and get good targeting. So this was, I think, the second project we tried back in 2013, 2014. We were trying to target ACAP12. Where's Mike? So Mike, yeah, so there's ACAP12, right? We were talking about ACAPs. and ACAP12 alpha to be more specific. And we actually got it. So we had a genotype. First of all, I'm not telling you all the day, we had multiple injections, I think like 6 or 8 injections. And finally, 1 out of 40 pups had both alleles. I'm sorry, the same allele with both LOXP sites, right? So you can imagine all the different permutations you can get. And you do get those. By the way, we did have one base mutation in one of the LOXP sites, but it's still cut anyway. All right, so this is one of those I love does anybody use bioRxiv? I love bioRxiv, look at it every week. So this is something that we put out on bioRxiv just recently, revisiting Rudy Janish's Cell paper, and this was sort of like a meta-analysis of multiple, like 50 groups, including our own. And it turns out, just you can read the sentence right there in glowing, it's horrible. Do not do it, do not do it, do not do it. You're better off doing something like this. So, and we've had good success with this. These are 2 examples here with the other 2 flavors of Calpona, CNN2 and 3, where we sequentially, so we put one in, and then we got our germline transmit, homozygosity. It takes a while. It takes as long to do this as it does ESL targeting, but it works. And so we've done this. But then recently, the gentleman whose name I can never pronounce, but thankfully he goes by Guru, which I can pronounce, he came up with what he calls easy CRISPR. And what it involves is a long single-strand oligo, and you can buy these now from IDT, so they're called megamers. I think they might be less than that now. They might be around 85 cents. Does anybody use megamers? Out of curiosity, nobody uses megamers. Well, did you know? So you can get megamers up to at least 2,000. I think they're even going up to 3,000 now nucleotides. Single-strand oligo. And very small, as you see, very small homology arms, like 50. I was astonished. I thought, no way. But if you understand even a little bit we know about repair during CRISPR, you'll get it. You don't need a lot of homology with CRISPR, folks. So the business about buying big oligos, you don't need to. I mean, Fung will say you need 30 nucleotides. That's fine. Because of the nature, because of resection, and then the annealing of the oligo. The oligo is a template for synthesizing the rest. It doesn't get incorporated as HDR would imply. Anyway, sorry. This works. This actually works. There's much better odds of getting your conditional allele with EC CRISPR than with the original RJ cell method. All right, so question, and we're coming down to the home stretch. Are embryonic stem cells even necessary anymore? I was really happy to hear that you still have ESLs here at Stanford. We still have ESLs. I think they're still necessary, and I'm just going to end with a reason why I think they're absolutely essential. So this was a project. I don't know if this is the right slide to start with, but so you all know now that our favorite gene is SRF. And we were interested in knocking it out in vascular smooth muscle in a conditional way to study it in the context of disease and so forth. Well, we never could do it, even with the gold standard Cre, which is myosin heavy chain Cre. We couldn't do it because of this phenotype. So you get this awful GI phenotype, and the animals die 3 weeks after tamoxifen. The longest lived ones. Many of them died before that, as you can see in this kill curve. All right, so we need a new Cre. All right, so here is a journal you've heard of, Gene, not a particularly high-impact journal. But we published this paper several years ago. I don't know, Vivek, if you're on this or not, but we were really intrigued with this result. So ITGA came out of a screen. Actually, it was the same screen, Mike, that our ACAP came out of. And we were really interested in this. So blazing in the aorta, low in the bladder and elsewhere, even uterus. Compared to SM22, aka TAGLN, it's much more restricted to vascular smooth muscle. And you can see then in the other data shown here that it holds up across species, including human. So this is a vascular smooth muscle restricted gene. And we thought, well, let's harness this and make a Cre. And we tried and tried and tried to make it with CRISPR and never got it, never got it. I finally said, okay, well, enough is enough. Let's just, Lin Gan is our core guy. He's phenomenal at doing this. He's made, I don't know, over 500 mouse strains, including the 30-odd we've done with him. And so we did the ESL targeting, and this has been a boon. And this is something that we're very close to submitting for publication. I've had many requests for it. I talk a lot. I tell people things in my posts. I'm like, what are you telling people for? Because I like to get the message out. And already we're getting requests for it. I'm happy to give it out. We're just doing some last-minute experiments, but this is really tight. And here's some data. So ITGA Cre is on the top, crossed with the MTMG reporter, okay? And then the gold standard is on the bottom. Now look here, bladder, and by the way, same setting, so we're not gaming this at all. I'm very strict about that with the people who make these images. So this is the same settings, the confocal microscopy, and you see that there's activity, but nowhere near myosin. And look in the intestine. Remember that phenotype I showed you? So it's almost not active at all in the intestine, which has been great because whereas before we were losing mice 2 to 3 weeks post-tamoxifen, we couldn't do anything. We tried to do an injury study, but we were getting like a wimpy and variable neointimal. It was horrible. We couldn't forget about athro and hypertension. This is 6 weeks out. Now we've gone 8 weeks out. Mice are completely fine. So this is awesome for things like SRF and, by the way, Lyamodin-1. Because remember, Lyamodin-1, which we're trying to make a flox allele with CRISPR, haven't gotten it yet, those would be predicted to have really strong GI phenotypes with the existing gold standard CRE, myosin heavy chain. So now we can circumvent that with this CRE driver. So we'll have this to give out very soon. So that was fast. But again, it's here for anyone who wants it. You can have it. Just don't publish on it. And utilize it. It's for you. Because I really, how many people are doing CRISPR in mice? Just out of curiosity. Even outsourcing. Is anybody doing CRISPR? Sean's doing it? A couple? Yeah, it's astonishing that everywhere, I always poll the audience. Even like plenary lectures, I poll the audience. Is why? I don't understand. I can't tell you how many times I've talked people out of making a cell line. Because I said, if you can grow the cells in culture, why not make the mouse? Then you get the cells, and you'll have a mouse. And several of them have done it. So anyway, it's an awesome technology, and this is the group. I got a small group. We've made over 30 mouse strains. Can you imagine a lab this size making 30 knockouts with ESL? It's never going to happen, right? But with CRISPR, it's pretty straightforward. So, and then I want to end here. Stanley, are you here? Yes? No? You have two people who I consider to be gene editing gurus, real gurus. These are the pioneers in genome editing. You'll recognize some of these names. Of course, the circled ones are here at Stanford. And really, I always like to end with this slide because I hail these people. I mean, this has just been phenomenal for my lab, my little mom and pop store of making mice. Our output has just like tenfold increased over the last several years. And I'm often accused of being an addict. And I'll confess, I'm a CRISPR addict. Okay, so we make these mice all the time. So I'll stop there. Thanks for your attention, and I'll try to answer any questions you might have.
Questions from the audience?
So I'll just say this. Why are you not doing mice? So at Rochester, it's within 5 weeks we can get the data, and it's $3,000. So some people outsource, and I don't get it. Like, the average is $20,000 and 6 to 8 month wait. Why?
So Joe, maybe I'll ask you about the bioarchive paper that you have, like, must have like 100 authors.
Yeah. One of those.
Rudy edited the paper. How did you manage to pull that off with all 100 authors and calculate the frequency is actually at 0.8% or something?
So this is Guru is the first author, and Gaten Bergio, you may know him. He's like a Twitter maniac. He's got a lot of Twitter followers. Those two sort of orchestrated it. I encouraged it because our experience was awful, as where it turns out many others. And so it was submitted to Nature Methods and it didn't get in. And one of the criticisms was that how did you standardize it? Well, there's no standardization, right? You got 50 cores. Some people use RNA, some people use RNPs, different concentrations, but we accounted for all that. So we have that in this. If you look at the bioarchive paper, you'll see that there are data that speak to those variances between groups.
Because it's already posted, did you get a reply from Moody's lab to see what's
Oh, he knows. Yeah, we've been in communication with him for years about it. I've talked to his injector, who's Coy. At Cold Spring Harbor every year, there's two CRISPR meetings, Cold Spring Harbor. Well, there's lots, but the two big ones are Cold Spring Harbor and Keystone. And every year at Keystone, we have a little satellite meeting of mouse users, right? And Rudy was there a couple years ago, and it came up, and he was pretty humble and pretty, I can't remember, the epigenetic state, and it was this hand-waving thing. I don't buy it, because in the cell paper, he was targeting MECP. For the developmental biologists, you'll recognize that gene symbol. And several of the groups targeted the same locus and tried to the best of their ability to do it in the same fashion he did, and zero. So we don't understand what happened, But clearly, it's not an approach to take. And I would, for those who are interested in conditional alleles and who isn't, I would seriously consider the EASI CRISPR approach with megamers. And that seems to work pretty well.
So questions?
It was so clear. Either that or you're like, okay, already, you told us every conceivable mouse you made. Sean.
No questions at all?
Sean.
Yes, Sean.
Yeah, Joe, that was a really nice talk, and it's a great tour to see how the different applications on CRISPRs that you have. I am curious, you did mention about how you eventually turned to use the ES cells instead because you couldn't do it out of CRISPR. What is it then about that locus? Did you ever figure out why it isn't so susceptible?
No, we tried every which way, and we got nada. It just didn't work. We tried one guide, we tried two guide, we tried double-stranded DNA, we couldn't try single-strand. Double-strand DNA with long homology arms, double-strand DNA with short homology arms. This was years ago we started this. This was one of the early projects as well. I finally just said, we got to get this Cree. The field needs it. And so it took the usual 1 year-ish to get it with ES cells, but we got it. But no, we weren't able to resolve it. It's interesting. So some loci, for whatever reason, you can call it just accessibility, maybe likely differential guide RNA activity. So we just genotyped a project where we had partial integration with two guides. And so the deal there is, I think, that one guide worked and you had local repair of, again, the oligo doesn't get integrated. I hope I made that clear. It's not like it's not HR. So I don't like HDR. So just that portion of the megamer was used as a template to repair that one spot, and then the other end didn't. So there's weirdness, and we still don't know all we need to know about DNA repair. So there is locus specificity. Some projects we get like half of the pups have editing, perfect editing, even with a deletion, perfect 3 base pairs upstream of each PAM, and then others not.
I asked because, we have like the exact same construct, try getting the same locus in multiple cell lines, and it's not all the ones end up getting insertions, for reasons that we all...
Have you played it all with, so sorry, we're going to go techno on you guys. So have you played it all with like NHEJ inhibitors or activators of HDR? The small molecules? Those don't work in mice, by the way. As my experience and others, they do not. So we don't use those, to try to bias it one way or another. Yeah.
So John, a separate question on the mice. Is there a particular strain of mouse that you go to, or have you noticed that different strains give you different efficiency? Yeah,
that's a great question, Joe. So we like Black 6 for reasons that would be apparent to everybody because we have so many models in that strain. But FEBs and SJLs work much better. They're much more efficient. I don't know why. I don't know why. And that's with pronuclear injection. Remember, I told you with pronuclear injection in Black 6 with the RMP, we compromise the litter size. We get better efficiency with the 3-component CRISPR, but sometimes we've had one project with just 2 pups. One of them had the right edit. That's 50%. So, but SJL, FEB, they're a good strain. But then you're looking at back crossing if you're going to cross it into another background, which is usually in Black 6. But there are known strain differences with respect to the efficiency of the edit. Yeah. Yes.
What are the challenges you think in organ targeting, specific targeting of the CRISPR technology?
It's doable. So like you're talking about making a conditional like point mutation. So I thought about that, but then I asked myself, well, why would I ever do that? Because if it's a germline transmitted allele, every cell has it, so why would I ever make that? But for reductionist science, you might want to have that. Oh, you're talking somatic editing in a specific way. Well, I'm immediately thought of Eric Olson's Duchenne muscular dystrophy project. He just had a science paper.
Stanford University.